Thank you.
So, good morning, everyone. Welcome to the Grand Rounds. And before I ask Neil Swerdlow to do a formal introduction to our speaker, I do want to personally welcome John here. You have been a colleague and collaborator for a number of years, working with Athena's group, with Mark Geyer, Stephanie Dulau, I think. And so you're kind of a semi-member here, and we welcome you. Neil, could you do the correct introduction now?
Morning, everybody, and welcome to our Grand Rounds. Dr. John Neumaier is professor and head of the Division of Psychiatric Neurosciences within the Department of Psychiatry and Behavioral Sciences at the University of Washington. John studied biology at Reed College and then completed his MD and PhD through the Medical Scientist Training Program at the University of Washington Department of Pharmacology, studying the pharmacology and electrophysiology of opioid peptides with Charlie Chavkin. John completed his residency and chief residency in psychiatry at UW, followed by a postdoctoral fellowship with Mark Hamblin studying molecular regulation of 5-HT1B receptors. Since then, he's been a true triple threat, maintaining an NIH-funded basic research program and outpatient clinical practice while running the psychiatry residency neuroscience training track since '94 and since 2015 as PI of their R25 funded research residency track. John's research has continued to focus on the molecular pharmacology of serotonin systems with primary clinical targets of affective disorders and addiction, but he's published on a wide range of topics ranging from DREADS to RTMS to striatal subcircuits, underlying decision-making strategies, a wide range of topics. He's received research awards from NARSAD, the Stanley Foundation, NIMH, NIAAA, and NIDA. He's a fellow of the ACNP, member of the ACNP Nominating Committee, deputy editor of Neuropsychopharmacology Reviews, distinguished fellow of the APA, and was named best supervisor by graduating UW psychiatry residents, among many other teaching and research awards. Today, John will talk to us about 5-HT6 receptors, location, location, location. Please join me in a warm welcome for our Grand Round speaker, Dr. John Neumaier.
Well, thank you. I usually say I would much rather give the talk than the introduction, and today I have that honor. So it makes me really nervous to do what you just did. So it's a pleasure to be here. I have a lot of long-term friends from the Serotonin Club in particular, but also my PhD advisor, Charlie Chavkin, trained down here. So I feel a sense of affinity already to the area and to my colleagues. And I have to say that Neil has become kind of a role model for me with having the R25 grant. He helped me kind of conceptualize how to run our program, but I think his is different, really creative, and so he's been really helpful. And I've had great collaborations with Stephanie, and I'm glad to be here today. I should mention, first of all, that the idea of location, location, location is not just that Seattle has now realized the importance of real estate. I know that it's especially important here, but I want to tell you about, and I want it partly to be a cautionary tale for those who are more junior in their research careers, that I've gradually wandered and meandered and started to understand the receptors I'm working on, but every 5 or 10 years I see them really differently. And rather than being ashamed of how poorly I understood them before, I think how exciting that I can wake up every morning and think the same types of receptors are still interesting. I'll show you a picture first of the lab group. Currently, one of the students who contributed to this in my lab was Dennis Smirnoff, who's an MSTP student here, but he wasn't in this particular picture. This was taken on Ugly Sweater Day, which is a new tradition in the lab. And I want to point out that this is not an ugly sweater, that my mother knit it for herself, but it just turned out 3 sizes too big. And then when I was 16, she said, well, I'd have to pay for the wool if I wanted it. And I held out, and I said, no way. And about a month later, she gave it to me. This is also a movie with jiggling, and I'm going to skip that part. And I see it. I know it's usually beautiful here in San Diego. I realize that. But it's sometimes nice in Seattle, too, and we actually put solar panels on the roof. And so we're making some energy up there. So it's not always raining, although this has probably been one of the wettest winters we've seen in a long time. Another connection that I have to San Diego is the mentor I worked with as a resident. I moved from working for Charlie Chavkin to working with Mark Hamlin, who did his MSTP here, and he was involved in the early cloning of a number of the 5-HT6 receptors. And this is a picture of most of the members of the 5-HT6. Oh, I think they're all in here now. There should be 14, and they can be organized into families based on their sequence homology and also on their signaling properties, although that, of course, now has become much more complicated, and I'll go into that a bit. And I've worked over the years on the 5-HT1B receptor quite a bit, a little bit on the 5-HT7, and a little bit on the 5-HT6. And the reason for this is that Mark gave me the plasmids. And when I was a resident, I thought I would try to take these molecular entities and try to put them back in the brain, try to use the molecular strategies that I was interested in learning to understand how these receptors worked in the circuitry, in a complex circuitry and in deep water. And the 5-HT6 receptor, we worked on it a little bit initially, but over time, I've worked on it more and more. And this was cloned by Mark, and it's positively coupled to stimulated adenylate cyclase through GS protein, although it's more complicated, and I'll get back to that later. It was an interesting receptor when it was first identified because when they did the initial pharmacological screening, they found that a number of the older antidepressants and a number of newer antipsychotics bound to this receptor with high affinity. And one, I wish I could remember where I saw this fact, but a majority or a plurality of clozapine binding, tritiated clozapine binding in the rat brain can be attributed to 5-HT6 receptors, especially in the striatum, as I'll get into more. Over the 5 or 10 years leading up to when I started to really work on this receptor, there had been a number of studies in rats showing, and in mice too, but mostly in rats, showing that the 5-HT6 receptor, when blocked, would improve memory or learning of very simple reward-motivated learning tasks. And people presumed this had to do with the hippocampus or the cortex, and that it had to do with memory consolidation. And I started to wonder about that. Let me show you an example of such a study. This is one that we did, a postdoc of mine, Siobhan Mitchell. And she did a number of interesting studies, and she was interested in going into pharma and has since done that and been very successful. And she wanted to do some drug studies in addition to the other work that I'll talk about more. And this is how she performed this particular experiment. It was a very simple memory task, whether the animal will remember a new versus, it'll remember an old animal that it's met before when it's challenged with a new one versus an old one. And so it's a social recognition task. We also did novel object recognition and that kind of those tasks as well. And if you put a control animal in, they will spend about 60 to 65% of their time with a new animal on a second challenge. So in the first challenge, they meet just one younger male rat, and then there are 2 in the second one. And you can see that this animal seems to be recognizing and preferring to explore the novel animal. And if we give that animal scopolamine right after it meets animal number 1 in the first session, then in session 2, they spend about equal amount of time with both animals, suggesting that they don't recall which animal was which. And if we gave this test ligand, which had 5-HT6 affinity, but also wasn't the cleanest drug, it had also 5-HT2A affinity, we saw a dose-dependent improvement in their recall up to the levels of what's seen in a control animal. We did similar studies using very old rats and other kinds of behavioral models. In the case of the old rats, we gave the drug for a couple of weeks and then took it off for a few days, and they still had much better cognitive function. So that seemed very, very promising. But I still struggle with the idea that people presumed that this effect was occurring in the hippocampus or the cortex, because that's where memory is. And I was aware that people had been using a task called auto-shaping, which probably wasn't precisely auto-shaping, but it was a very simple action outcome reward task, and 5-HT6 ligands would work in that task as well. So I started thinking about, well, where are the receptors? I was also concerned that these drugs weren't that selective and there weren't any good agonists at this point. And so since we had been using viral-mediated gene transfer to manipulate 5-HT1B receptors, I thought we would swap the 5-HT6 receptor in. And we were packaging these in my lab using HSV. Not a perfect system, but one that's been helpful for us. So this is where the receptors really are. If you look in humans or if you look in rats, you have very strong expression of the binding sites and of the messenger RNA in the caudate, putamen, ventral striatum. And you do find this receptor in cortex and hippocampus, but it's intensely expressed in the striatum. And so that just really struck me. And I thought that we ought to focus on that brain region. And I can't tell you how naive I was about where behavior came from in the brain. I just knew that I should go to where the receptors were. And at the end of the session, maybe you will believe me when I say that I'm naive, because it's true. But then I further considered where in the striatum should we focus. And I didn't know the difference between ventral and dorsal striatum. But one of the graduate students with Dan Dorsa, who's an MSTP student at UW, had done immunohistochemistry for 5-HT6 and peptides of the direct and indirect pathway. He found clearly this receptor is expressed in both pathways. So there's a location and a location. And later I'll tell you more about the third part of this connection, which is that eventually I appreciated that 5-HT6 receptors localized to the primary neuronal cilium in neurons that express it. But it's an interesting target for pharmacological development because it's so CNS specific, because it has an interesting potential profile pharmacologically. And there have been some phase 2 and 3 trials looking at one drug that Lundbeck has developed that at least initially looked like it would enhance the outcome with people with early stage Alzheimer's when they were also put onto cholinesterase inhibitor. It was an add-on trial. My understanding is that a more recent trial hasn't been so successful, but there are a number of very selective compounds out in the environment. Okay, so that's one issue. And then here's another issue. Here's a distribution pattern of the receptor binding, I believe it is. I can't see from this angle, in a rat. And so you can see strong expression in the striatum. And if you look in a human, it's very similar. And the ventral and dorsal striatum and putamen have high levels of expression, much higher than elsewhere. But most people these days would study a mouse, and one thing you might do is make a knockout mouse. And Larry Teacott made a knockout mouse, but it was a mouse with very little phenotype. They did about 20 tests, and one of them was significant. And that was the end of the paper, and really the end of his work on that receptor. But I think we now understand why that had a very modest phenotype. This is the pattern of expression in a mouse. It is expressed, but it's expressed at low levels, and it doesn't show the regional distribution. And in addition to that, the pharmacology of this receptor has diverged in mice from what you see in rats or humans. So if you look at a correlation of the affinity of various drugs for the rat or the human 5-HT6 receptor, it's very tightly correlated. But if you look in mouse, there's a lot more variability. So there continue to be studies in mice, 5-HT6 receptors, and this issue is never really acknowledged in those papers. And I think that there's much to learn from mice with 5-HT6 receptors, but they're probably not a great model for how they work in rats or humans. So we did a number of studies in the early years with Siobhan Mitchell taking the lead, and she expressed a hemagglutinin-tagged 5-HT6 receptor in the dorsal medial striatum, and Susan Ferguson did it in the nucleus accumbens. And they did studies that suggested that 5-HT6 receptors, when increased, would interfere with the animal's learning a simple task or even a Pavlovian association. And we found that we could block this with an antagonist, that we wouldn't see it with sham receptors, and it mattered when we expressed the receptor. And I'm not going to go through some of that early information. I'm going to show you a more recent study. But Siobhan continued to think that it's probably affecting the recall of how to do the task, because the way we ran those early action outcome experiments, it took several days for the animals to learn to press the lever. And so she tried giving the 5-HT6 antagonist at the end of each session, but it wouldn't reverse the learning. And I convinced her to try it before the sessions, and that did reverse the impairment on the learning that the 5-HT6 receptor overexpression displayed. So our next study was done by Dan Eskenazi, who's now a fourth-year resident at Columbia. And I suggested to him that he could change the way he did his action outcome learning paradigm so the animal would learn in a single session. And he did so in either control animals that were expressing GFP or animals expressing 5-HT6 receptors in the dorsal medial striatum. And he found that indeed the animals with increased 5-HT6 receptor learned the task about half as quickly as the control animals. Once the animals have learned the task, you don't disrupt behavior. So it really had to do with the learning. And I'm not an expert in learning, but we did do quite a few experiments. We mapped it in the different parts of the ventral striatum. We looked later at habitual behavior, and we found out what everybody already knew, which is the part of the striatum you study matters. And when we moved our viral expression away from dorsal medial striatum into the center of the striatum, because we thought that would be nice because there'd be equal space on both sides. And with a little OCD, that's a very good feeling. But the experiment wouldn't work anymore. And then we discovered that it really mattered where you expressed it. And then we went and looked at dorsolateral striatum. We found out that's where the habitual behaviors were affected. And nucleus accumbens had more to do with learning of a condition-place preference for cocaine, for example. So at that point, I was convinced it was learning, and no direct effects on reward. But I mentioned the idea of the direct and the indirect pathway. And meanwhile, Susan Ferguson in my lab was developing viral vectors and she was interested in studying cocaine mechanisms using dreads, which we did. And I was interested in making these vectors so that we could manipulate 5-HT6 receptors in the direct or the indirect pathway. And as is often the case, Susan's idea not only was a better idea, but one that has had more impact. And so she used viral vectors that targeted the direct pathway with a pro-dynorphin promoter-driven HSV to manipulate receptor or anything we want, and with the indirect pathway with the enkephalin promoter. So in this case, though, I'm going to focus on 5-HT6, and we've been able to increase 5-HT6 receptors in the direct pathway or in the indirect pathway. Now, why would we do this? My thought at that time was, and I was finally aware of how the direct and indirect pathway had segregated dopamine receptor expression, at least partially segregated. And I was unaware of this when I started this project. Another humiliating admission I'll make this morning. And so I started with dynorphin and enkephalin simply because I thought the expression levels would be much higher than a receptor, and it would be a better promoter to use. I didn't really understand all the work that had been done earlier to understand the segregation of gene expression in these pathways. But if the D1 receptor was in this pathway, the direct pathway would tend to be activated by dopamine. Whereas if the D2 receptor, an inhibitory receptor, was in the indirect pathway, it would tend to be inhibited by dopamine. And I did get that, and that dopamine was probably causing differential activation of the pathways. And serotonin, which is mostly ignored and gets very little respect in the striatum, even though it's present, the 5-HT6 receptors, as well as the 5-HT1B, are in both pathways. I don't know if the levels are the same. I have never seen anything regulate 5-HT6 receptor mRNA in the striatum, but 5-HT1B is very dynamically regulated. If you stress the animals or give them cocaine or withdraw them, all kinds of things will change 1B expression very dynamically. So our thought was that if direct and indirect pathway were differentially activated, then having 5-HT6 receptors in both pathways tended to interfere with that differential activation. And you could say that would make the animal more stupid, but I would say it makes it less inclined to acquire new habit-related learning. And so it slows down the acquisition of the automated behaviors, such as we all do every day. If any of you drive a stick shift around the beautiful curves on the coast, you've learned how to listen to cues in the sound of the engine and the vibration, and you make shifts without thinking at all. And that requires your striatum to learn to do that kind of thing. And 5-HT6 receptors perhaps would interfere with your acquiring behaviors too quickly. I'm sure that's overly simplified. So the next study we did, and I'm going to focus a good deal of time on this, it was recently published, and it was done by 2 graduate students. Matt Brodsky is now graduated and is working with Eric Turner as a postdoc. Eric was formerly down here. And Alec Gibson is an MSTP student in the lab. And also, they worked under the direction of Sunil Neumaier, who's a former postdoc, who's now an assistant professor in our department, and is associated with my lab, but has her own funding and her own projects, and is very helpful for me because she understands so much more than I do about cocaine self-administration. In this study, we expressed viral vectors in either the direct or the indirect pathway using the pro-dynorphin or the pro-enkephalin-driven viral vectors, which I would say are 90% selective in a rat. They're not probably as good in a mouse. And we expressed these in the nucleus accumbens shell, where there's strong expression. And then we waited a while, and then we did a pretty complicated experimental design. We started with FR1 cocaine, and then as the experiment unfolded, we started adding things to the design, trying to understand what was going on, which is not perfect. And in fact, in the future, we're going to change some of our manipulations or our behavioral tests to try to learn more. But I'll show you these experiments now. We did FR1s for about 11 days, I think, and then a few days off, I believe. And then we did some progressive ratio sessions, and we looked at breakpoint analysis, and then we did a dose-response curve. And at the end of the experiment, we did some locomotor activity just to see if there were some non-specific effects of behavior, which there weren't. When we expressed the 5-HT6 receptor in the direct pathway, we saw no change. We saw no increase in the learning or really any change at all. And that's shown in both of these top curves. On the first few days when the animals had increased 5-HT6 receptors in the indirect pathway, they took less cocaine by about half. And I expected, as the study proceeded, that they would catch up, and they simply were having slower learning, as we had observed with sucrose pellets in previous studies. But they never actually increased to the same level as the animals taking cocaine with the control virus, the encapsulin GFP. They take about half as much cocaine. And that's at an FR1 schedule. So I'm not going to tell you the experiments in the order that we did them, but maybe just in order that's a little easier to absorb. We did a dose-response curve. This was these animals were trained on a moderately high dose of cocaine, 0.7 milligrams per kilogram. I wouldn't do that again. We did. And so we looked at lower and higher doses, and we simply didn't see any differences in the direct pathway 5-HT6 receptor animals. But in the animals with increased 5-HT6 receptor in the indirect pathway, they took less cocaine, particularly at the lower doses of cocaine, and by about half. And we saw this whether you look at the total cocaine taken during the session or the number of infusions. I think it was a 2-hour session. You can see better than me. A 2-hour session. Okay, so then at that point, I said, why don't you try to look at the cocaine administration during the session? And we learned that we had been using a legacy program on MedPC that really didn't capture the information very well. So a lot of time was spent trying to capture when the levers were pressed because that wasn't coded properly. And if you look at the animals with increased 5-HT6 expression, you see what rats do when they take cocaine. They load up when you first put them in the cage in the morning. They take a burst of activity, and then they go into a second phase where they take cocaine at a more moderate but very regulated pace. So this is regulated cocaine taking, something that George Koob taught me once was not interesting. That what would really be interesting is out of control cocaine use. I completely agree with him, but you have to start at the beginning. So I'm okay with that. But when we looked in the animals that had increased 5-HT6 in the indirect pathway, we saw much less cocaine taking at the beginning of the session, and then they took less, but especially I think that the noteworthy change was at the beginning of the session. And this little bump here, this is due to a slight hiccup in our programming too. They had a couple of minutes off where they maybe missed one lever press. That's unfortunate. We've now we've sanitized all of our MedPC program. The other thing I want to point out is that when you put a control animal in this box, it takes cocaine in, half a minute or so, give or take. But the animals with increased 5-HT6 were waiting a minute and a half before they started taking cocaine. I don't understand this. I would think that they are in a relative withdrawal state and that they would be enthusiastic to start taking cocaine. Well, one idea might be that they aren't in withdrawal. Another idea might be that they're very ambivalent about taking cocaine. They want it, but there are aspects that they don't want. There's something about what they expect that's negative. And the other thing I've wondered about is if they have some defect in the way they tell time or update their mood state. Now, I can't prove that with any of these experiments. It's just something I'm thinking about for the future. We took this data with the assistance of Dave Roberts. We used some modeling it's been out for many years, but I've seen it more and more recently, where you take the time of the cocaine infusions, and based on a bunch of prior work, you can estimate the tissue concentration of cocaine based on that timing and dose of cocaine self-administration. We found that the control animals and the 5-HT6, I think this is a control animal from the encapsulin side, they take about twice as much cocaine as the animals that have increased 5-HT6 receptor, as I mentioned earlier, but they also seem to titrate around a different tissue concentration. And my colleague Paul Phillips suggested perhaps they're reaching satiety, and they find a lower dose of cocaine perfectly reinforcing, but they don't like higher doses of it for some reason. And we looked at different doses with this modeling, and it seems especially at the low doses there's a big difference. So the last thing that we wanted to do before completing the study is to try to get a better sense of their motivational state. And with the data that we had, without starting over again, which is what Dave Roberts suggested that we do, we had progressive ratios data. I mentioned we did that early, and there were really no differences at all with progressive ratios in any of these groups. And I should mention, these groups were run entirely separately. They were run as separate experiments. So you do see some differences between the two experiments, but I think that's just the normal variation that occurs. But the problem with progressive ratios is it looks really at cocaine taking at the end of the session. And I told you that these animals were having a difference in the beginning of the session. So at that point, I suggested, what if we did conditioned place preference? That's how I got started in the cocaine field. And so I learned that if you give a very low dose of cocaine and you have a very brief pairing, you're likely only to get a conditioned place preference in animals that are very sensitive to the cocaine reward. And so we used 5 milligrams per kilogram, which is a pretty low dose, and a 15-minute pairing, which is tuned for picking up reward, but at a pretty low dose. And by moving the time of the pairing, you can switch to a conditioned place aversion, and we've done this in previous studies. So what we found was that the control animals did not develop a conditioned place preference for cocaine under these conditions, but the enkephalin 5-HT6 animals had a robust conditioned place preference to this sub-threshold dose of cocaine, suggesting again that they were more sensitive to the rewarding or reinforcing properties of the drug. So that's where we stood at that point, and it made us think that we were deconstructing the contribution of 5-HT6 receptors in the brain, and that there was a balance normally in the direct and the indirect pathway, but when we created an imbalance, we could detect that if we did it in the neurons that had D2 receptors in it that were prone to be less activated by dopamine. And so I think the 5-HT6 activation through serotonin is probably compensating for that and interfering with this differential activation. That's been the theory. And we're continuing to study it in other ways, some of which I'll touch on today. We are using some dread receptor studies that are parallel to this in some respects, and I'll go into that too, but not directly parallel. So our theory then is that we've uncovered an aspect of this the differential gene expression in the direct and the indirect pathway. Now I want to shift gears. At this point, I started to have some concerns about HSV, which I think has been a great system, but has some limitations. And my colleagues in Seattle, Tom Nasco, I was on his general exam committee and his supervisor committee. I was watching him use some retrograde viral vectors with great elegance, and so I was inspired to do that as well. We started packaging CAV2 CRE in my lab and combining it with AAV viruses that had a floxed transgene, DIO format, and that's shown here. And we started this, as I mentioned, we started this with DREADS. We still haven't done it with 5-HT6. And once CRE acts on this AAV construct, it can flip the DREAD into the orientation where it can be expressed in cells. And here's a picture of some neurons where, in the nucleus accumbens core, where we put the retrograde CRE into the substantia nigra area. And you can see the expression of some DREAD receptors. And so this is an approach that we're using in a grant that just got funded after many submissions. I wore them down and they funded the project. And I'll tell you about that in a minute because I think it's fun. It shows you where my lab is going. It also shows you my lack of focus. I would appreciate what you said I had focus because nobody else in Seattle thinks that. We also have been using these strategies in lateral habenula with DREADS and with ribotag. Here's an example of a sagittal picture showing dorsal raphe expression. We've decided to mix a little bit of CAV2 ZS green in with our CAV2 CRE because it makes it really easy to see where your injections are. You can see this almost without fluorescent light sometimes. And so we add, at this point, 2% ZS green virus to our CRE virus. And in this case, it tracks back into the lateral habenula neurons and it flipped the DREADS And you can see that in red in the blow up of the lateral habenula sagittally. So I mentioned the dreads earlier, and I'll just remind those of you who are not familiar with them. These are receptors that were devised in Brian Roth's lab, and they were mutated so that they would gain the capacity to either excite or inhibit cells through different flavors of dreads. I'm being pretty brief about this for the sake of time. And it's a very elegant system, and very low levels of expression are really needed in order to get a very nice dread-induced change, which is somewhat different from what you encounter with optogenetics. Overexpression hasn't really been too much of a problem, and there's very little evidence of constitutive activity with these receptors. There's been a few reports, but we haven't been able to detect it. We've looked. So we used dreads in the same system I was just describing in this experiment for the pilot experiment for the grant that finally got funded now. This is an example of animals that were taking cocaine on a 6-hour schedule, and they have an escalation of cocaine self-administration, another contribution from San Diego, this model. And at that point, we do 2 tests. We do a very short reinstatement type test under extinction conditions on day 1, just for 30 minutes. But then the animals are simply allowed to be in an abstinent state for several weeks, and we're going to do it more about 35 days in the study that we plan. But in this experiment, we did it for 25 days, and we gave them a dose of CNO, either or not, right before they were tested. And what you see in a control animal, and it may be surprising to some of you, is that control animals over the first month of abstinence will show an increased propensity to seek drug and depress the lever. And this is referred to as the incubation of craving. And we found that when we inhibited the direct pathway neurons with dreads in the nucleus accumbens core, we were able to blunt this seeking considerably. And this is simply a pilot experiment. I've shown this slide for a while, and now we're going to start to really pursue it more with our experiments. And we would like to do something similar with 5-HT6 receptors, but we're not going to overexpress them anymore, and I want to explain why and how I discovered what was wrong with our method in more detail. Okay, so let's go back to 5-HT6. I've told you the third part of location is the idea of the cilia. A couple of my colleagues in Seattle, Dan Storm and I'm blanking the name now, the other person, I apologize. They said to me, you know, the 5-HT6 receptor that you study is in cilia. And they would say this at lunch or a coffee break at a symposium, and I'd say, okay. And I had no idea what they were talking about. And then about a year later, I had a light bulb that this was profound, and I had been ignoring it, and it was right under my nose, which is another lesson I want to point out today, that I've done this many times. I think all experienced researchers will describe having this experience of having all the clues right in front of them, but not putting them in the right order so that they would appreciate what was being suggested by the data. So I thought all of a sudden, that's really remarkable, because this is not part of the synapse. And if you had strong activation or inhibition of receptors in the cilium, it's not going to completely disrupt synaptic information flow the way dopamine receptors can, or glutamate receptors, or GABA receptors. It's going to have a more subtle modulatory effect. And what is serotonin all about? About being subtle and about being modulatory. And this 5-HT6 receptor is the only serotonin receptor that gets into the cilium of the serotonin receptors. Let me show you a little bit more about that. The primary neuronal cilium is formed by 9 microtubules that are not surrounded, that they have nothing in the center to cross-link to. So they're non-motile. They don't generate motion, but they do flop around, and they're flexible. And they're absolutely packed with signaling molecules. And some people refer to them as the antenna of the cell. Most or all cells have a cilium at some point during their lifetime. If you, and it emerges from the mother centriole, and one of the reasons cancer biologists are interested in cilia is that if you can make a cell have a cilia, it's not dividing anymore. And so they think cilia are good. I've grown to also like them as well. But neurons don't divide. So neurons have cilia at a high rate. About 80% of the time, 80 to 85% of the time, we can see a cilium cross-sectionally in striatum. And my guess is that that's a representation of the fact that they have a tendency to adapt over time. As I'll show you in a minute, they can change in their length over a matter of hours. And they're very dynamic. And I know very little about cilium, so I've added the list of things I don't know. But it is interesting that receptors are tethered to a mechanism that's like a trolley that carries proteins up or down the cilium. And there is a mechanism for restricting access. This transitional zone restricts access to proteins that aren't supposed to be in the cilium. And although there have been reports that D1 receptors and D2 receptors can get into the cilium, I don't know if they always do, but they can. But 5-HT6 receptors are in the cilium, I'll submit, and they shouldn't be anywhere else. And this is true about a variety of other receptors which are poorly understood and may be great targets for drug development. Here's a picture of an electron microscope, a micrograph showing 5-HT6 staining and a cross-sectional profile of a cilium. And here's some pictures we took recently to support the confocal grant that I'm supposed to have into the university to be mailed this afternoon. We'll see about that. You can see that the 5-HT6 is strongly expressed here, and in this case it's co-localizing with adenylate cyclase 3, which is a cilia-specific adenylate cyclase. When Dan Storm knocked that adenylate cyclase out, the animals have profound learning deficits. And this is a cross-section under super resolution, showing kind of the donut-shaped expression of the 5-HT6, the HA-tag 5-HT6 receptor. So what are cilia? Why do they matter? Mostly they get studied by developmental biologists. And ciliopathies is what you call a disease where the cilia protein has been mutated. And it leads to all kinds of birth defects, but they commonly involve problems such as polydactyly and kidney cysts or liver cysts, situs inversus, and cognitive impairment is very common. And Joubert syndrome is one of the more common forms of this. There's a fellow in Seattle who studies this, so I'm getting to know them. But nobody pays much attention to what do they do in the adult. They're still there, and nobody paid much attention. So I'm showing you here some 5-HT6 staining in the neuronal cilia of striatal neurons in vivo. And you can see adenylate cyclase marking the cilia on the left. It's hard to see, but there's the 5-HT6 receptors in many, but not all of the same cilia, and they overlay very nicely. We got a hold of the 5-HT6 knockout mice from Larry Teacott. Even though I argued that they have imperfections, they do have 5-HT6 and they are in cilia. And we saw no 5-HT6 staining. This is not a good antibody, and that's one of the problems in the field that I struggle with, and that's why we like to use HA-stained receptors when we can. And we did the same thing in vitro, basically. We learned to do primary neuronal culture so we could try to test some of the hypotheses about signaling that we were developing. And although, for now, I'm going to talk about endogenous 5-HT6 receptors. And we found that when you treated them with an agonist, nothing happened. And that doesn't entirely surprise me because it's hard to get rid of serotonin in a culture condition. That's always my suspicious thinking. But the other thing is that this receptor has been reported to have constitutive activity many times. And I tended to not believe that in vivo, but at least I have some evidence in endogenous expression that might be true. But if we give an antagonist, it reduces the length of the cilium by about half. And this is typical. Drugs that activate adenylate cyclase and cilium tend to make them longer. And so blocking that seems to make them shorter. And this happens with a perfect pharmacology in terms of the affinity of the antagonist. We've used other antagonists as well. It happens over about 12 hours. So these change pretty rapidly. Okay. So then we started to transfect receptors into primary neuronal cultures. And here you can see the 5-HT6 in the cilium. Oh, this field didn't work the way I wanted to. Okay, that's fine. And we marked the cilia with AR13B in this case. And there's the overlay. What you see down here is another neuron that was also transfected with 5-HT6, but look, it's everywhere in the cell. It's overexpressed. And we didn't really worry about that initially, but we quickly started to worry about it a great deal. And I'm going to talk about that a little bit more. Now, I built the slide backwards. I apologize. I was going to try to show you just that first. So we became very concerned that most of the work with heterologous expression of 5-HT6 receptors in the last 20 years has been done under conditions where the 5-HT6 receptor is not in the cilium. And that could lead to a lot of problems. First of all, we found that if we transfected the cells with very small amounts of 5-HT6 plasmid, then we tended to get levels of 5-HT6 cilia, which were pretty close to what we would observe in endogenous receptors. This experiment is done in wild-type cells that express 5-HT6 receptor. But the higher the level of plasmid, 5-HT6 plasmid you include, the more it tends to escape out into the cell body. And it may still be in the cilium too, but it's hard to find the cilium when you have a big green neuron. We found the same problem in knockout cells. So we tried to perform our experiments at low levels of transfection. And at different times in different people in the lab, it use slightly different amounts of it. Well, why does the 5-HT6 receptor go to the cilium in the first place? Other people who've been working on this question have predicted that there's a 5-amino acid consensus sequence that's present in most receptors that go to cilia, some kind of a trafficking signal that it's the secret handshake to get into the primary neuronal cilium. So the first thing Matt did in this is he mutated the 5-HT6 receptor by deleting it. And similar to some work by the Nicotin lab, he swapped the 5-HT7 loop into the 5-HT6 receptor. They're both GS-coupled serotonin receptors. And what we found was when we made one of those two mutations, we reduced the amount of 5-HT6 receptor that localized to cilia by about 75%, not 100%. And we've not been able to completely disrupt distribution of the receptor to the cilium, which makes us think there are probably additional motifs within the protein structure that allow it access to the cilium. We wondered if this was only happening because they were being expressed in, originally we were doing this in wild type cells. We thought maybe they're dimerizing and the wild type receptors are dragging the mutants in. That doesn't seem to be the case because even in knockouts we see that. Okay, so shifting to the work of, for the last little bit, of a postdoc in the lab, Adam Leigh, they noticed or we saw the literature accumulating evidence that 5-HT6 receptor had lots and lots of different signaling pathways. GS-coupled adenylate cyclase for sure, but also fin kinase, interactions with CDK5, with mTOR, and with other proteins that are structural features of the cell. And this has become, it's been expanding rapidly. And in all of these cases, people are overexpressing the receptors, and sometimes in neurons, often not. And we became a bit skeptical and even a little bit of a conspiracy theorist that this was wrong. And so we wanted to take a look at that in our system. And so we made a couple of mutations. We made one that wasn't exactly what we intended, just by an unfortunate error. There are several described 5-HT6 mutants. One of them blocks the ability of serotonin and other ligands to bind to the receptor. It still shows that constitutive activity. There's another one that blocks GS coupling, and so it can block adenylate cyclase activity. We thought we had that one, but it turns out we didn't. So we're going to do that in the future. A third mutation is known to interfere with the interactions with fin kinase, although we can't replicate the fin kinase in our lab so far. But we've made this mutation. And then Adam has used knockout cells and these constructs and transfected them at a very low concentration into neurons, primary neurons. And he also includes a MAP2B RFP, so you can see the dendrites rather easily to quantify dendrites as well as cilia morphology. And he did a curve of expression. And if you look at the percent of the neurons that have 5-HT6 in the cilium, it really peaks around 12 to 15%, something like that. Why is it so low at these lower concentrations? It's probably because there are just too few neurons that got transfected with it. So I think that's an artificial low number. But as he increases, you can see most of the receptor ends up in the cell body. He can't even detect it in the cilia in those cases usually. And in these experiments, he found that the cilia length was, from knockout neurons, was increased when he expressed the 5-HT6 receptor, whether or not it had one of these mutations. In all cases, it seems to be longer. And because we didn't have that GS-dead mutant that we thought we had in the study, the sequencing turned out wrong. We think that this is mediated, the cilia elongation is mediated by adenylate cyclase activity. That's the general feeling in the field, but we haven't completed that yet. But interestingly, we replicated what other people have observed is that if you look at dendrite outgrowth, that we see an increased dendritic outgrowth when 5-HT6 is overexpressed, but if we block fin interactions, which I think also blocks a lot of other things that haven't been considered, like beta-arrestin, I think will be interfered with, that this doesn't work anymore. So it's possible that fin signaling or something relating to pairing with molecules at the C terminal of the receptor is accounting for this effect. So that is interesting, and it's possible then the 5-HT6 receptors in the cilia are remodeling the structure of the neuron somehow. I'm not going to show the data today because we've only done a couple of days of electrophysiology, but my hippocampal electrophysiology neighbor tried some 5-HT6 drugs on wild-type hippocampal neurons from rats, and it really changed the excitability of the neurons tremendously. An antagonist was able to reduce the excitability of the neurons remarkably, but they didn't do too many slices, so I don't really have much data on that. So at this point, what questions do we have? Well, we know the 5-HT6 receptor should only be in the cilium. And overexpression, I believe, was informative. I believe my earlier results, but I don't believe they were optimal experiments. And other people have told me that for a long time, but now I believe them and agree with them. And so in my next grant, it'll be a refutation of my last two, which normally you would want to go and hide under a rock. But I think that I try to wear my continual learning state on my sleeve, and that's not a bad thing. So we want to be able to get 5-HT6. We want to look at 5-HT6 receptors only when they're in the cilium. And we would like to look at their signaling. We wonder if they regulate the excitability and the morphology of adult neurons. But we especially are interested if they're a way of modulating behavior in animals. There hasn't been any focus on cilia receptors in general in adult pharmacology that I'm aware of. So it could be what we learn about 5-HT6 will inform what we might learn about other receptors that are also in the cilium that might be even more amenable to drug manipulations. So at this point, my students told me we should do CRISPR. And I think I said, how many of you know it in the room know what CRISPR is? I'm not sure if I do. I still get, it's very mysterious. So CRISPR, I will explain it. If you really want to understand it well, I'll put you in touch with my MSTP student who understands it more deeply and has been working with it. But the basic idea is that you are co-opting an immune system that microbes use to recognize foreign RNA. And you have a guide strand which you can design to match any gene of interest. If you get the Cas9 or one of the Cas9 enzymes into the cell, it will find the DNA and it will make an edit, usually of a deletion, possibly even insertion, depending on how you design them. And this is where he has the details and I don't. I said RNA before and I meant to say DNA, excuse me. So the target is DNA. And we thought, why don't we try to target 5-HT6 in rat brains and see to what extent we can efficiently remove them. I don't think we have to hit every single 5-HT, every cell to have a positive effect, but we'll learn and we'll find out. So he got an F30 grant funded recently to start this work, and also to try to knock out adenylate cyclase 3 in the same cells to try to get a better handle on is it working through adenylate cyclase 3 or not. And these are thoughts that I really want to develop into my new R01 when I get some time. We have other ideas as well. So this is Alex's work. He's an amazing student. And perhaps when he goes to hit the research trail, I'll encourage him to come down here. Like my other student from the lab who's thinking about psychiatry, I would prefer you leave Seattle because I think there's lots to be learned somewhere else. I also wonder if we might be able to introduce small mutations like I was showing you earlier in vivo so that we can block signaling one way or another. Perhaps if there is a beta-arrestin effect, which we're working on, can we block beta-arrestin signaling? And the other idea I had is, since I told you the antibodies aren't very good for this receptor right now. So what if we did HA tagging in vivo of targets of interest? Everybody in the room who does lab-based research probably has a protein target that they can't reliably detect. I think it's worth, and you don't have to get everyone. You have to get enough of them to be able to study them. And so I think that might be an interesting strategy. And my student, when I suggested it to him a couple weeks ago, the next day he came and said, it's already been done, but they did it with GFP, which is a little surprising because GFP is a much bigger insertion. So, but in the brain. So this is how we're planning to do it. We're intending to use a DIO cassette to control the expression of Cas9, and then a second transcriptional cassette to express the guide strand. And Larry's Whiteful is about 3 months ahead of us on these things in his lab. And so far, they're having lots of success. We've been able so far to develop a regular non-flux version for beta arrestants, and that seems to be working really well. So I think we're on the road. Our plans going forward will be to try to knock out or mutate the 5-HT6 receptor in vivo and try to find out what happens when you mutate this receptor, not in the germ cells, but later on after the brain is already normally developed. I think that's really important. I didn't mention it, but there is some evidence that 5-HT6 receptors, when absent from birth, that it has a different effect than when they're absent later in life. So we want something that's more of a conditional knockout. And I'd like to, and he plans to do these experiments using cocaine self-administration, but we'll use more Dave Roberts' ways of thinking about how to get a better handle on how this receptor and the different pathways is affecting the motivation to take cocaine. And I'm hoping that it really ultimately will have some impact on developing treatments, harm reduction treatments that might reduce an individual's propensity to take high doses of cocaine. It would be progress. And of course, we're interested in what it does to the cell's morphology. So just to conclude, I think 5-HT6 receptors, I've learned a lot over the last few years, and I've revised my thinking. And I think that rather than saying I was wrong all those years, I think I'm learning something new and taking different angles. And I want to encourage those of you still in training to look at your oversights as opportunities. And I think that the 5-HT6 receptor may be a really interesting target for more than just cognitive processes. It may affect more subtle things about how the striatum works, where it's expressed so heavily, perhaps for hippocampus and cortex as well. And we would like to take some very discrete steps that I'm not going to repeat again to try to understand this. But I appreciate your attention today, and just want to thank all the different support that I've had for this grant and the people who've worked on the project. And I'm finished. Thank you. Who knew?
What on earth are they? I thought they were all on paramecia or
Right. They're only there if you stain for them. It's like basement membrane and glia. You know, there's no glia in the brain unless you stain for them. So.
Well, thank you very much for this wonderful presentation. And you're willing to... Would that be okay?
Sure.
Sure. And I'll come around, use the mic, hold it close to your mouth, speak clearly, succinctly.
Hi, Abe. I already knew Abe was going to ask a hard question. So.
No, I don't think it'll be a hard question. But so I'm still thinking about the slide. It was maybe a third of the way in where you had the animals that were not taking the cocaine nearly as quickly. So not only would they take less cocaine, but the latency was about 3 times greater.
We saw that with sucrose too. When we did dorsomedial striatum, the animals on the first session, one way of looking at acquisition of learning is how long does it take before they put it together and make the first lever press that's spontaneous, and it's dramatically elongated when you overexpress 5-HT6 in the indirect pathway.
Even for sucrose, huh?
For sucrose.
Okay.
You know, and that's the first time, so that argues against the aversion idea. But
yes, right. That's where I was going to go.
True, though.
Okay, interesting. And I guess the other thing I was going to ask about that is, should I still be thinking about those studies, or because you were overexpressing the receptor in a way that would get it out of the cilia, should I have a big asterisk in my mind about the phenomenon?
Well, that's the question I don't know the answer to. I think that everything we saw made sense, and it parsed in a pathway-specific manner. So I think we learned things about the pathway. And I'm not sure the answer yet. I think what we would really have to do is compare low versus high levels of expression on the behavior. And that won't be easy, but I suppose that could be done. One thing I've thought about doing, for example, although I told you this receptor is already constitutively active to some extent, people argue. If you made a constitutively active receptor, you could express that, and you could compare it, and it could still be in the right cell compartment, because you wouldn't be changing its level of expression. Although, who knows what else you'd be changing. So, yeah, I mean, I'm just being honest with the data that I think that's a limitation. But if we each look 10 years back on what we did, we'd do it differently.
Yeah. No, I appreciate the honesty throughout your talk quite a bit.
Going back to the work on the cocaine, it reminded me of the work that Bob Post had done years ago, where he found that female rodents required half the dose of cocaine to get the same behavioral effects. And I'm wondering if you have evidence of that.
We haven't done any cocaine self-administration in females yet. We've been doing stress studies in females and mice in a very different project. And so I'll say now because, and maybe I'll have to retract it in a week, our first look at RNA-seq data from serotonin neurons is that we get dramatic changes in the translation of RNAs in serotonin neurons of females, much more so than males. And we don't know if the important thing is happening in both or mostly in the females. We don't understand it yet. But I'm kind of excited about that. Our program officer required us to add females before it was mandated, but it's turned into something really interesting. And so I appreciate that she did that.
So I was just wondering if you have any functional readout of how the 5-HT6 receptor being expressed in the cilia versus the rest of the neuron really affects the neuron. Can you look with electrophysiology or something?
That was the whole idea of those mutations, that we're going to restrict the receptor away from the cilium. I had an R21, right? When I recognized all this, the first thing I did is write an R21 with just about no data, but I had been thinking about it a lot. And it was well constructed, but our hypothesis that we could prevent the 5-HT6 from getting the cilia was rejected so far. I still think there's more to it. I think it could be beta-arrestin. There's evidence that beta-arrestin is important in cilia trafficking. And if we were to disrupt the beta-arrestin interactions that have yet to be shown, we're trying to do that right now. It just there are too many projects. We haven't gotten that done fast enough.
Right.
So I think I would like to do what you say and to try to compare them. And when we made this 5-HT7 loop 5-HT6, we thought that was going to be a great mutation for this because they both have similar activity and they'd be expressed similarly, but it just didn't work. So I don't know the answer. We also tried to make some biosensors that were cilia specific, and that just didn't go very far because it was hard and it wasted a lot of my postdoc's time. So I wish in retrospect we hadn't tried to go down that avenue.
Yeah, I guess one of the big questions is whether, how much of a difference it's actually going to make, in terms of the...
My prediction is that you get too much signaling when it's outside of the receptor, both in the magnitude of the signaling, but also it might start talking to systems that it shouldn't. But each one of the systems that's been implicated, I think, have been found in cilia too, at least under some circumstances. So it might be that all of that signaling is also valid. We don't know.
Mm-hmm.
Were you going to mention putting the CRISPR system using the 2 virus, or did you mention that in the last?
Oh, yeah, I didn't explain myself well. So it's a 2 virus strategy where we're going to use the retrograde Cre going into either the ventral pallidum or the substantia nigra or VTA, depending on how we're going to do our nucleus accumbens injections. And then we'll put the floxed and inverted CRISPR construct into the nucleus accumbens. But this will become modular, and we can flop in any little piece. We'll be able to replace the guide strand with anything very quickly, just with Gibson assembly. And the student is named Gibson, so he's really good at it too.
Well, it's pretty amazing because you'll never need to make another floxed mouse.
Well, that's
if you've
true.
got that working, you'll just be able to change the viruses.
With some of the faculty here, I was lamenting one of my program officers is so disappointed with me these days. And one of the things she's been always disappointed is my 1B knockout mouse, which was meant to be amazing and really cool and better than Renee Hen's, and we never got it made. And so now I'm thinking I'm still going to pay that debt.
That'll be even better than the mouse.
Yeah. That mouse was still going to turn green or red, depending on whether it's been knocked out or protected. So it was cool, but it didn't work.
So, tour de force, I want to go back to the very beginning where you enticed us about the importance of these receptors because they're may be involved in the mode of action of antipsychotics, antidepressants, pro-cognitive drugs, and showed us some data on the novelty measures. So you've told us about cocaine self-administration, reward mechanisms. How does this cilia receptor system fit into this larger profile of psychopathology?
Well, that's a great question. I mean, I'm not sure if it fits into the pathology. It might fit into the therapy. And so I think of it as the back door, the password to the computer system of the cell. Maybe we might be able to manipulate things through the cilia while we're not doing so much damage to the synaptic information flow. As far as depression goes, Svenningsson thinks the 5-HT6 receptor is really important for depression. But I think it's that he's looking at striatum in ways that engage depression models, whereas I've been engaging cocaine. So I think it's possible that his results are exactly true. He's been doing it in mice, which I think is a limitation. We recently, in our RNA-seq I mentioned, my postdoc said to me, and you know what showed up and was differentially expressed? 5-HT6. And I said, it's not supposed to be in those neurons. That really, because I've probably told, as a reviewer a few times, I've shot papers down that talked about 5-HT6 autoreceptors. And I maintain that they're not autoreceptors, they're expressed in the GABA cells next door. But he found that in males, stressed or unstressed, you can barely not detect it. The level is just exceedingly low. And in females, in the unstressed animals, exceedingly low. But it went up like 5 or 10-fold, some crazy amount in the stressed females. And this is very small and preliminary. We haven't validated it yet. But we did a second run that was a slightly different treatment, and he saw it twice. And so it could be that 5-HT6 receptors are potentially recruited in some states that nobody's ever looked at before. And if that were the case, you might want to block them, which is what all these drugs do. And the ones that I should mention, clozapine, olanzapine, are drugs that sometimes people recognize pro-cognitive qualities with. I've seen it. It doesn't always happen. And whenever, if any of you have ever seen somebody blossom from Haldol to olanzapine, in addition to any metabolic consequences, sometimes it's really remarkable how they'll improve. And so I wondered if 5-HT6 had something to do with that, because most of the typicals don't bind to 5-HT6. There's a few that do some. I can't remember which ones off the top of my head. Oh, well, I think they're also in hippocampus and cortex, but I would say the receptor is always a slave to the cell it's in. And I would say further, perhaps, it's always a slave to the neighborhood it's supposed to be in. And that if we lose sight of that, then we will make mistakes that 10 years later we'll realize.
Okay, any other comments, questions? If not, is that oh, you're getting ready to clap. Okay, all right. Okay, so thank you very much. And first of all, since you're already informally in the Department of Psychiatry, we'd like to present you with a Department of Psychiatry baseball hat. Since you put on solar panels, it may be that you should wear a hat when you go running, jogging, walking your dog, whatever.
Thank you. I'll try it on. I'm getting a spot that needs covering, so that's good.
Okay, great. Thank you very much.
Thank you.
Great.